Hello, Dr. Magori.
Hey, guys.
You ready, Liz?
Yep.
Well, in that case, I'd like to welcome everyone and thank you all very much for coming to the first biology department seminar of the quarter. We're going to have seminars every 2 weeks at this time throughout the winter and the spring quarter, so you are invited to all of them. Today, we're going to be talking about the science around the COVID-19 vaccine with Eastern's own Drs. Louis Matos, Jason Ashley, Javier Ochoa-Reparaz, and Krisztian Magori. So each of them are going to talk for about 10 minutes, and then we're saving time at the end for questions. If you think of questions during any of the presentations, there is the Q&A feature that we'll be using for questions after Dr. Magori's presentation. So feel free to enter your question in there. And with that, I'm going to turn things over to Louis.
Well, good afternoon, everyone. So yes, I'm going to talk about SARS-CoV-2 mRNA vaccines, what's in the vials and how did it get there. And here we go. And so if you think about it, what are the goals of a good vaccine? Of course, it should be safe, but then it also needs to be effective. And in that way, it needs to deliver some sort of an antigen. In this case, the vaccines we're talking about are going to be delivering pieces or the information for making pieces of the spike protein on the coronavirus. It then needs to elicit an immune response, and finally, it needs to generate memory. And so the goal here is that our bodies learn to recognize the virus without actually ever meeting said virus. It's just going to get exposed to a small portion. And so, of course, we now have 2 vaccines approved in the US, the Mrna vaccine and the Pfizer-BioNTech vaccine. And these both are mRNA and lipid nanoparticle vaccines. So what the heck's a lipid nanoparticle, I guess would be the question. And we usually refer to them as liposomes, and these are cationic lipid particles. And here you see just a small assortment of the wide range of lipids that Mrna has patented. These liposomes are about 80 to 180 nanometers in diameter, and this is a fairly good depiction. We have a phospholipid bilayer, and in the middle we have a cavity where the payload, if you will, can be delivered. And these are really useful for a wide variety of payloads. And here we'll have the RNA in there. And so what is RNA? And so just as a reminder, here we have DNA as our double-stranded old friend with hydrogen bonds keeping the phosphodiester, the sugar phosphate backbones together. And so of course here then we have the single-stranded RNA, which has the bases available for interaction with ribosomes, as we'll see. Here on the left, I am showing the image that Mrna put into its patent. Each of these numbers means something. And so on the right here, I have another picture showing what it actually means. So we have the 5' cap, which protects the 5' end, a small untranslated region, a signal peptide, which is going to help the dendritic cells determine exactly what to do with this peptide. The particular gene of interest, which in this case is again encoding a portion of the spike protein, a 3' UTR, which is an untranslated region that will not make protein, and then a bunch of A's, the poly A tail, which protects the 3' end in addition to doing a few other things. And so how does this work? So generally we think about it, we have DNA, the DNA is opened up and we transcribe it into RNA. And then the RNA is translated by ribosomes into proteins. And I kind of thought of what I thought was a fun analogy. And so if we think of our DNA as kind of an encyclopedia of quilting patterns where every pattern is a gene that could make a protein. And then if we open up this and get a page, we have a pattern, and that pattern is a particular gene. And if we give that as mRNA to the ribosome, we have these busy hands, the proteins of the ribosome will actually make a polypeptide. And it's that polypeptide that then can go and generate an immune response. And so what the vaccines are doing is instead of going to the DNA, we're short-circuiting the system and putting in directly the mRNA. So we give the body the specific sequence and no viral components at all. And then the body produces the particular part of the protein that we've determined to be most immunogenic. And so how does this actually work? Well, here we have our RNA molecule. It gets put into the liposome. Here we see it in the middle. The liposome is taken into the dendritic cells by endocytosis. Once the RNA is released, the ribosomes pick it up. The proteins are transported, they're exposed on the outside, and then the immune system can generate the antibodies and memory. So I thought it would be fun. So here's the Mrna patent, and the name of it is Modified Polynucleotides for the Production of Secreted Proteins. As you might imagine, these patents have been written as broadly as possible so that they can use this delivery system for as many things as possible. The patent is actually 240 pages. No, I did not read all of it. As part of the abstract, which is in patent terms, is the claim, a plurality of lipid nanoparticles between 80 and 160 nanometers with RNA encoding a polypeptide is their main claim. And here we have the biotech patent, particles comprising a shell with RNA, again, really broadly written. And this one, RNA-decorated particles, preferably to RNA-decorated liposomes. And so it sounds like their process is not as specific. It is not generating only liposomes, but partial molecules that also can carry on the RNA. And so what's actually in the vials? And so for the Mrna, it's RNA encased within the delivery vehicle. So literally it's just RNA and lipids, and of course the buffer. For the BioNTech, we have RNA-decorated liposomes, so RNA, the lipids. However, even though not mentioned in the claim, they also have bisphosphonate molecules as adjuvants. And one example of that I've put here is zoledronic acid, which has actually been used for a long time in osteoporosis therapy. And so as far as the production of the mRNA, we can basically write the gene however we want it, have it synthesized, clone it into a plasmid, have it grown, and then do transcription and just make lots of RNA, cap it, and eventually purify it through. And what happens here then is that we have highly pure messenger RNA with nothing else in there. And then we can take that and we can encase it in liposomes. And I've shown here just one general process where you can sonicate the lipid with the payload, in this case, the RNA, and then through an extrusion process, you can actually make the liposomes. Once you have that, you need to actually sterile fill. That's a really critical part of the whole process. And we actually have a major sterile filling center here in Spokane. They actually do synthesis, but Jubilant Hollister Steer is a really large biotech company here in Spokane. And they can fill several million vials a day, just to give you an example. But basically, if everything is working, that guy just stands there with his hands in the air in a sterile suit. And if any of this sounds interesting, you could think about doing a Bachelor of Science in Biology with a biotechnology option at EWU. And please contact me if you want to learn more about that. And just this morning, some breaking news. AstraZeneca, their vaccine was approved by the European regulators. This is a different type of vaccine. It's vectored vaccine. Basically, I like to think about it as a harmless, neutered adenovirus. So they've taken away all of the viruses all of the genes that will lead to an infection. It delivers the gene to the host nucleus, and then the host produces the mRNA and the protein, as we discussed earlier. This is very safe. This technology has been used for quite a while. And the nice thing is it's actually cheap to make, and it's stable at 4 degrees. And so it has some advantages over the mRNA vaccines. And some more breaking news, Johnson & Johnson's vaccine is 66% effective. And, so they'll probably be seeking a special permission to use it soon. And Johnson & Johnson has lots of capacity. So that's going to be a nice thing, capacity for making vaccines. and they've already promised 100 million vaccines by June. And so with that, I thank you, and I now hand it off to Dr. Ashley.
Thank you, Dr. Matos, for that very interesting introduction. It's a very exciting vaccine technology, this BioNTech and Mrna vaccines. If they prove to be effective in the long term, it might give us some opportunities to vaccinate against a lot more diseases than we've been able to vaccinate against previously. One of the questions that has come up with these vaccines is the speed at which these vaccines have been developed and given emergency use authorization. So some questions that I was thinking when just perusing news stories was, for this BioNTech vaccine, how was it tested? Was there anything in the experimental protocol that diverged from what is typical for new drug assessment? And is there anything about that that might give us cause for concern? So I looked at a couple different things. I decided the best way I could answer this question would be to compare this mRNA vaccines testing to a previously approved vaccine and just see how those studies compare to each other. Hence, we are going to discuss two different vaccine trials over the next 10 minutes and hopefully get to some of the answers to these questions. Vaccines are tested more or less the way every drug is tested, in a series of phases. There's a preclinical phase where the investigational drug is assessed in vitro and in animal models. And then the numbered phases correspond to testing these drugs and vaccines in human beings. The goals of each phase of the study are overlapping, and they are mainly differentiated from each other in terms of size. As you go from phase 1 to phase 4, more and more participants are enrolled in the trial. Safety and efficacy is assessed throughout. Really, in phase 1, that's simply a very small pilot study to find what doses of the drug are tolerated. So these are sometimes referred to as dose escalation studies, where a small number of participants get different doses of the drug and they are monitored for reactions to it. Once you get to phase 2 and beyond, efficacy begins to be considered. So phase 2 is where you begin to understand whether or not this is efficacious. And phase 3 begins to increase the sample size to determine whether or not rare events in terms of safety are occurring and whether or not the efficacy that you've previously established holds. If the FDA is satisfied with the data coming out of the phase 3 clinical trial, they can approve the drug for public use, but that is not the end of the testing process. Phase 4 is a post-approval period where patients that are now receiving the therapy are continuously monitored for continued efficacy determination, as well as detection of rare events. So in today's talk, we're going to compare a phase 3, phase 2 study of the BioNTech vaccine to another vaccine, and that vaccine is Gardasil. So you may or may not have heard of this vaccine before. This is a vaccine against human papillomavirus. It causes warts on the skin, and it's also one of the leading causes of cervical cancer. So if you can vaccinate against HPV, you can theoretically vaccinate against cervical cancer. So the original Gardasil was approved in 2006. It was a quadrivalent vaccine in that it contained vaccination against 4 different strains of HPV. It's been retired. It's been replaced with another form called Gardasil 9, which vaccinates against 9 different strains of HPV. Of course, the vaccine that Dr. Matos introduced to you it's got a really snappy name. It's BNT162b2. I'm just going to call it BNT for the duration of this talk. And this is a vaccine against SARS-CoV-2, which is the virus that causes COVID-19. So you already know how it works. It's an mRNA vaccine. Let's take a look at our sources. So most vaccine trials are ultimately published into the scientific literature. So anybody with access to the paper can read the study and look at the details for themselves. So these are the 2 papers that describe the phase 3 trials for these 2 vaccines. The Gardasil trial was published in 2007, which was quite a bit later after the trial was completed. It ran from January 2002 to March 2003. The BNT trial was published much more closer to it being completed. It ran from July to November in 2020. So one of the first differences you might notice is the Gardasil trial ran for over a year. The BNT trial only ran for a few months. And as we go ahead, we're going to find out why that's not as big as a problem as you might at first assume. So if you're going to test this, you need people to test it in, and you design your study to recruit patients that are relevant to the disease you're trying to prevent. So the trial that tested Gardasil was called the FUTURE trial. A lot of times these trials are given fun names. So the FUTURE trial is the Females United to Unilaterally Resist Endo-Ecto-Cervical Lesions. So the goal of this study was to reduce cervical cancer. So the recruitment group for this were young women aged 16 to 24 that were not pregnant, because that might alter the safety assessment of the vaccine. And also they needed to not have a history of abnormal pap smears or genital lesions, because these were going to be endpoints of the study. The BNT study was much more broad. The goal of the study was to prevent the spread of COVID-19. So anybody age 16 or older was potentially eligible for this study, provided they were not pregnant and they had no prior history of COVID-19. The median age in the group was 52. It was a little under half female. One big difference you might notice is these were both phase 3 clinical trials, but the total number of enrollees in the Gardasil was just over 5,000. Total number of enrollees for the BNT was over 37,000. Big difference of this, a lot more money was involved in the formation of the BNT trial. As part of Operation Warp Speed, the US government was offering some supplemental funding to drug companies to develop these vaccines. Pfizer turned that money down, but what they got instead was the government promised to buy at least 100 million doses if their vaccine worked, at a tune of about $2 billion. So with that guarantee, Pfizer was willing to put the money forth to make a much larger study. So half the group received placebo, and then half the group received the drug, which is typical for a clinical study. So the endpoints for the two different drugs were related to the diseases they were trying to prevent. In terms of Gardasil, they were looking for reductions in genital and cervical lesions. And they were also looking specifically as to whether or not the vaccine was protecting against the strains of HPV from which it was designed. For the BNT, they were looking at COVID-19 cases throughout the study, particularly looking at least 7 days after the second dose of the vaccine. And they were doing testing on these individuals. So they were checking both symptomatic and asymptomatic cases. So anybody that was infected earlier, they were included in the study, but they were analyzed separately. So they were also looking specifically at severe COVID-19 as well as safety events. So anything classified as an adverse event would be a bad thing associated with the treatment, mainly breaking down to injection site events, systemic events, and then serious events, which would be things requiring hospitalization or death. So let's take a look at the results for Gardasil. If you've never seen one of these before, this is called a Kaplan-Meier curve, and it demonstrates over time the number of people that achieved a particular endpoint. In this case, this is going to be genital lesions in this example. So here you're looking at the placebo and the vaccine, and over the 3-year period of the study, you can see that the placebo steadily rises and the vaccine stays relatively flat. Of course, within 6 months, the first 6 months, there were actually more people who'd been vaccinated that got the disease. It took a year for the vaccine and the placebo groups to begin to differentiate from each other. Not surprising, HPV is a sexually transmitted disease, so level of sexual activity is of course going to limit the exposure to the virus, so it would take a longer time to reach some of these study endpoints. All told, the vaccine is about 95% effective against preventing lesions caused by strains of HPV that the vaccine was targeted against. Now let's take a look at BNT. Same curve, but take a look at the time scale. These are days. The placebo group and the vaccine group differentiated from each other within 10 days of the study. So COVID-19, it's airborne, much more easy to spread than a sexually transmitted infection. So you don't necessarily have to wait a full year to know if you're starting to have a protective effect. So running out through 112 days, we see a very impressive reduction in COVID-19 cases among individuals that received the vaccine. So between the doses, it was about 52% effective, but greater than 70 days after that second dose, about the same level of effectiveness as Gardasil. But because it's a more transmissible disease, you find out faster. As far as safety goes, this is broken down into adverse events and then subclassified as serious. For Gardasil, there were more injection site events with Gardasil than placebo, but systemic and serious events looked about the same. For BNT, if you looked at any adverse events, there were more adverse events with BNT than placebo. And this corresponds with some anecdotal stories that have been coming out. People feel bad for a little while after they've taken these vaccines, but then they get over it within a couple days. If we break that down into serious events, not a very big difference between the groups, nor were there a big difference in serious events were causing people to withdraw from the study. The published study itself says they'll continue to monitor safety for 2 years after that second dose. As far as regulatory statuses go, Gardasil was approved in 2006 based on data from this study and then a larger follow-up. And then, of course, the original Gardasil was replaced with Gardasil 9 in 2014. For BNT, technically it's not FDA approved. What it has is emergency use authorization. And in order to be evaluated for this, there must be safety and efficacy data from either a completed phase 3 clinical trial or one that is ongoing. And then it must also have safety data of at least 2 months of follow-up, which this study had, and more than 3,000 vaccine recipients, which this study also had. So all told, what we find with this BioNTech vaccine is the study was conducted pretty much identically to a previous vaccine trial with a shorter time scale related to the increased transmissibility of the disease and with a safety profile that is within the acceptable range of the FDA. So with that in mind, here are some additional reading for information if you like, and I will turn it over to Dr. Ochoa-Reparaz, who will explain what happens to the vaccine once it's inside your body.
Great. Thank you, Dr. Ashley. All right, and hello, everybody. I hope you can see my screen. So today, my section is going to be focused mostly on the immune system and the responses that the vaccine, within the vaccine, is triggering once we get injected with it. So I'll give a brief introduction on the immune responses that we would need in order to trigger protection against the virus. We will talk a little bit about general aspects of goals of the vaccines and the responsibilities that vaccination should carry with, and how they work, and what's next in terms of research. And I picked this, it's a cartoon, it's an old cartoon of Edward Jenner, who's associated historically with the process of vaccination, being the first scientist using vaccine as a way to protect against infection. And I picked this cartoon because you can see people, individuals here with these weird faces. Here you have a face of a person with a cow. And that's basically highlighting the fear that in those days, vaccines triggered in the population based on not knowing how they worked. Because Jenner worked with cowpox, and vaccines developed based on, on animals in order to treat the individuals, and people thought that they were going to get cow or bull features in their faces if they were vaccinated. But that was just based on basically unknowledge or not knowledge. How do we trigger immune responses to vaccines, to viruses first? Well, we all have what we call an innate immunity by default. We are all born with a rapid way to fight against pathogens. It's very unspecific, it's always the same, and it does not trigger memory. Now, it's made of physical and chemical barriers, like these walls and water around here. We have resident sentinels, phagocytic cells, and all the cells that are always there. We produce cytokines and molecules that can block and neutralize pathogens. But again, this is all very unspecific, and it doesn't really last too long, and they don't generate memory. Sometimes, though, it is enough to get rid of the pathogen if there's not enough viral virulence associated or not enough viral load. That might be sufficient. However, we know that that's not the case, at least for SARS-CoV-2 and COVID-19. So it is important for us, in order to get protected to trigger more advanced, faster, and specialized responses that are dependent on what we call the adaptive immunity. It takes longer, it takes about 10 days or so to trigger, but it's very specific, and more importantly, it generates memory. It's mediated by cells that can kill directly the pathogen and by antibodies, which would be produced by plasma cells. And we're going to talk about both mechanisms today. First, the use of antibodies to fight viruses. Antibodies are produced by plasma cells, right? And they bind to pathogens, not only viruses, in this lock key system, basically. So they are very specific to certain molecules that pathogens have, okay? B cells or plasma cells that produce antibodies that were originally B cells that matured into plasma cells produce a number of different antibodies. Now, I'm going to focus on two of them that are particularly important in the context of COVID-19 and the protection against SARS-CoV-2. One of them is IgA, and it's important because it's a neutralizing antibody. By attaching these antibodies to the spikes, and Dr. Matos talked about them, of the particle, the viral particle, we avoid the interaction of these spikes with the receptors that the virus uses to enter the cells. That's why IgA is important. And then we have IgG, which is another type of antibody that is basically able to tell other cells, other immune cells, to find the virus and kill it, okay? And obviously the process is a lot more complex, but for us, at least at this point, it is important for us to acknowledge the importance of both IgA and IgG antibodies. But that's not the end of the story. And sometimes when we focus on the studies that have been published in the context of COVID, we focus mostly on antibodies. But viruses are also killed by immune cells, and vaccines also trigger cell-mediated killing responses against pathogens, including viruses, okay? And we're not going to talk about the mechanisms, but basically we have two large populations of cells that are specialized in killing other cells that have been infected with a virus, okay? So again, cell-mediated responses are also important to protect us against SARS-CoV-2 infections. Now, so let's go now to the vaccines. Well, two goals mainly, and Dr. Ashley and Dr. Matos already talked about them. The first goal is obviously to protect against the infection. And the second goal is to generate memory. That way, we will be protected against a later infection. But those are the goals. How about the responsibilities? The vaccine needs to be safe, it needs to be effective, but also needs to be easy to handle, easy to transport, to store, to inject, it should be affordable, and so on. This is important because even if the first vaccines that have been developed against COVID-19 are effective and are safe, we should continue doing research in order to facilitate all of these characteristics that vaccines should have. I just put here this graph of an example of a given another disease and the importance of vaccination in the number of cases in thousands over the last 30, 40, 60 years or so, and how dramatic the drop is once the population gets vaccinated. All right, so how are our immune cells able to sense and see what the vaccines, in this case, the mRNA vaccines, exposed? What we see is only the spike, as Dr. Matos mentioned, our cells are going to express the spike protein, nothing else about the virus, okay? So in that sense, we are safe. This is what our immune cells will see. And the spike protein is a protein made of peptides, and each peptide is made of amino acids, right? And each of these big protein contains fragments that in immunology, we call antigenic determinants or epitopes, okay? And these are the little fractions that our immune cells can see. And different antibodies will bind specifically to different epitopes or small fractions of this big protein. This has relevance for what we're going to be talking about later today, and the new variants of the virus and the potential effects of vaccines against those variants of the virus, okay? So how is this happening? Well, let's imagine this viral spike that we administer, and that is going to be sensed, recognized by immune cells. We call them dendritic cells. And dendritic cells engulf, fragments of dead cells, debris, even the spike itself, and it basically takes it, degrades it into small little fractions, okay? So it's a process of enzymatic and pH digestion that makes it little fragments, little protein fragments that are then presented on the surface of the dendritic cell using specialized molecules that I'm not going to talk about, okay? Each protein or each fragments will be presented by maybe different molecules that are, again, responsible for these presentations. This is represented here. Imagine this is the cell surface of that cell, the dendritic cell, okay? And here you have those molecules that I'm not going to talk about that. These three fragments are coming from the same protein, three different epitopes or antigenic determinants. Now, they are coming from the same protein, but they can be recognized later by lymphocytes, important cells of adaptive immunity, with different epitope specificity, basically. These molecules will bind to different fragments of that big protein. And once that happens, those cells grow in numbers, they proliferate, and they form clones. And each clone has a specificity that is determined by the epitope that they recognized. This clone is specific for triangles. This clone for the circles, these clones are specific for, or this clone is specific for the squares. What we are doing is we are creating clones of lymphocytes that are specific for certain fragments of the same big spike protein, okay? And more importantly, adaptive immunity triggers memory. So a fraction of these cells will persist, will live longer with specificity to these little fragments of the same spike protein, okay? Those T lymphocytes, once they grow, we basically can separate them into two big groups. We call them helper cells if they are this type of cells, CD4 T cells, and we call them cytotoxic T lymphocytes if they are CD8 T cells. They have different functions. The helper cells are called helper because they help other cells, including B cells, to perform a function. In B cells, these helper cells help the B cells to produce antibodies, okay? And they also help phagocytic cells to be better destroying infected cells. Now, the CD8 T cells or the cytotoxic T lymphocytes are ready to kill infected cells once they have been activated in that process. Now, the other lymphocytes that are important when we discuss vaccines are B cells or B lymphocytes because they produce those antibodies. And at first, they always produce one, IgM, which is a good antibody, but it's not perfect. It's not, the affinity of these antibodies are not great. They bind, but perhaps worse than other types of antibodies, okay? In order to generate better antibodies that bind better to those fragments of the protein, we need the help from these CD4 T cells, okay? And once that happens, those B cells can produce that important IgA. Remember, it was neutralizing. It was able to neutralize the interaction between the virus and the cell, and IgG, which helps other cells to directly kill the pathogen, the virus. All right. So is this happening naturally? Yes, it can happen, but it takes about 2 weeks. So what we use or we do with vaccination is allow to have memory lymphocytes already that have been going through this process before. That way, when we get exposed to the real virus, these memory cells get reactivated and they start directly killing the infected cells or directly producing the good type of antibodies, the ones that have better affinity, the ones that are basically better performing their functions. Not only that, we make them faster and we make them in higher quantities. That's the beauty of vaccination. We have a set of cells that are ready to act against a pathogen as soon as they see it again, okay? Without having to get exposed to the pathogen first. So how long does this immunological memory last? Well, we don't know yet because the studies and the vaccines have been approved very recently. However, there are already some studies showing that at least we have immunological memory for 8 months after infection. So we hope that vaccination will trigger a similar or even longer memory, but we still don't know. Another important question: should we wear a mask after vaccination? The answer is yes. Why is yes? I told you that vaccines are good in inducing the production of antibodies, but it looks like the IgA production is more efficient than the IgG production, is more efficient than the IgA production. So the vaccines are very good controlling the process of, or the replication of the virus once they have entered the cell, because those cells can be killed by, cytolytic cells. However, these vaccines might not be really or completely effective producing IgA, and we know that IgA is the one that neutralizes antibodies. So it is possible that a vaccinated person, even though it's not going to get sick, might be able to still spread viral particles that person has in the respiratory tract. We still don't know for sure, but it is a possibility. Okay, and so even if we get vaccinated, I think at this point it is acknowledged that we should still wear a mask. How about the new variants? Will existing vaccines also work? Important question. We actually have some answers today. Okay. No trials have been done before until something came out earlier this morning, okay? We still need to get more studies in vaccinated individuals that are exposed to these new variants. There were some in vitro work suggesting that the ones that have been approved were effective against the new variants that are now happening. These new variants have little mutations in some fragments of that big spike protein. However, remember that the same protein can trigger different immune responses because of those different fragments, those epitopes. So perhaps we don't get a full protection, but we might still get some partial protection, okay? And this is what the study that was, at least this news press, I didn't read the article yet, but there's one vaccine that delivers about 90% efficacy against COVID-19 in the UK. However, it's a lot less potent in South Africa. Okay, so that perhaps it might be related to the variant that is running wild in certain parts of South Africa. We don't know that yet. Okay, more research is needed, as I mentioned. This is one of the adenovirus vaccines that have been developed, as Dr. Matos mentioned. 1 dose needed. The cold chain is not that restrictive. It is safe. Okay, however, we still don't know if it's going to be fully protective or no, but the Johnson & Johnson vaccine, the phase 3 clinical trials were released, and it looks like there's some disease protection variability depending on the location where the vaccination occurred. US, 72% protection, Latin America, 66%, South Africa, 57%. So there might be some variations in the protection, but nevertheless, it would be great news to have a third vaccine approved, and my guess is that this is going to happen pretty fast or pretty soon. Anyways, that was it. Few minutes of pure immunology. If you guys have more questions about immunology, or you want to take it in the spring or in the summer, just send me an email. We also talk about host and pathogen interactions in the winter, or we will talk about it next winter. So please send me an email if you guys are interested in taking this class or these classes. Thank you. And I should mention that now I'm just going to give the podium to Dr. Magori, Krisztian Magori.
Okay, can you see my screen? Okay, good. So I will briefly talk about the numbers behind all of these things, being the biostatistician myself. So first question is, do we still need the vaccine? Or is it like, mostly everybody had COVID-19 at this point and it doesn't matter? Well, it's not the case. We still need the vaccine. So on this graph on the upper right corner, you see the epidemic curve for Spokane County from the regional health district. And so in the past week or so, we had lower case numbers, but just yesterday we had about 500 confirmed cases reported. So it's not like COVID-19 is going away. There was a little bit of downturn in the left-hand side that you see after our latest spike around the holidays, but we're not out of the woods yet here by any sense. And I'm sure you heard that Spokane County is still staying in phase 1. And part of that is because our positivity rate is still 20%, meaning that 20% of everybody who gets tested for COVID-19 is positive. So we still have a lot of COVID-19 out there. We certainly need the vaccine. This pandemic is not over yet by any sense. What can we expect? This is a projection graph from the Institute of Health Metrics and Evaluation in Seattle by Professor Chris Murray, and he's updating this pretty regularly. So the dashed line is today, and this is showing the estimated daily counts of cases. And so on the right-hand side, you see what into the future, and there's a couple of different scenarios. The projection one, the one that is sort of brownish, maroon, that is sort of what we can most expect, is that the number of cases are going to go down towards the summer. But that's assuming that everything is going well with vaccinations. The purple one is when we have these variants in, and they might be spreading more efficiently, and so that's why there's probably more cases. And I want to highlight the red one, the one that says worst case. The reason why that is so much worse is because people who have been vaccinated in this model are assumed to go back to living normal. So it doesn't have to do anything with variants. It's just that people who have had the vaccine, they now think they are safe and they don't need to wear masks and they can socialize. But as Dr. Ochoa-Reparaz said, they are still potentially transmitting the virus, even though they themselves are protected from serious impacts, and that could make the pandemic last longer and be worse. So that's sort of an interesting thing to note. How is vaccine distribution going? So in the US, so far about 50 million doses have been distributed, but only about half of those have been administered. Many of those are en route. The leftover 50% are en route to the states as we speak. Some of them are sitting on shelves because they were put away so that people can get their second dose. And some of them are sitting on shelves because there weren't enough people to vaccinate in the particular category that they wanted to vaccinate at that moment. Just a little comparison with other countries. On the left, on the lower side, you see the per capita, per 100 people, how many vaccines were administered. Israel has been doing really well, way better than the United States. And even the United Kingdom has had a faster speed. But other countries like Italy are doing a little bit worse. And sort of nationally, Washington is doing kind of the same as most of the other states. There are some states that are doing much better, like Alaska and West Virginia, for some reason, has been just excellent in terms of rolling out their vaccination distributions. So why is it important to get enough people vaccinated? So one thing is to protect yourself, right? So that we all understand that. We don't want to get COVID-19. We don't want serious health complications. But let's say you're thinking, well, I'm healthy, or maybe I already had COVID-19. Why should I still get the vaccine? So the reason why you want to get it is because of herd immunity. And herd immunity is a situation where you have enough people vaccinated, and that protects other people who cannot get vaccinated, such as children under 16 years old for whom these vaccines are not yet licensed, people with immunodeficiency, or people with prior allergic reactions that should not get the vaccine so that they don't get another reaction. But how many people do we need to vaccinate to have that situation, that sufficient herd immunity? So that depends on R0, which you might have heard of this number before. It's the number of secondary infections produced by a single infectious individual in a completely susceptible population. If we can increase herd immunity by vaccination, we can bring R, the effective R, that is currently the reproduction rate of the virus, below 1, and then the epidemic will decrease and eventually the pandemic will go away. The critical proportion that we need to vaccinate can be calculated by this simple equation. And we know what these are for several other diseases. Let's say for measles, it's 83 to 94%, but measles has an R naught of 12 to 18. So what is the R naught for COVID-19 or SARS-CoV-2? There are various estimates. The one that I picked here is 5.7 with a 95% confidence interval between 3.8 and 8.9. Doing the calculation, that would give us a proportion to vaccinate of roughly 82% and maybe a 95% confidence interval between 73 and 88%. How many people would that mean from 330 million people in the United States? That would be about 271 million people. That's a lot of people to vaccinate. It might take a while. I found this graph from Yongung Gu's website, who is another modeler who looks at COVID-19. And this is what he calls the path to herd immunity, basically a projection of how fast we could ramp up vaccination rates and also just develop herd immunity with a combination of vaccination and past infection. And so basically he's guessing that by the end of summer, September, we might get around 70%, 75% immunity to the virus. But whether or not that's enough, that depends on that might not be quite enough. I know Dr. Fauci talks about 85% usually that we would want to aim to get. So it will take significant amount of time to still reach that. I'm sure many of you have seen this graph, which is the phases of vaccination in Washington state. So we're still in tier 1A, tier 1B now, and hopefully soon we can get into the subsequent tiers. But people who don't have any pre-existing conditions and who are under 50 years old and don't live in congregate settings, it will not be until the summer, or maybe for some of us even the fall, before we can get these vaccines under current circumstances. Now, if the new vaccines come in, the Johnson & Johnson and the AstraZeneca, those might flood the market with a whole bunch of more vaccines, and that this situation might become faster, and that's our hope. So what happens if we don't achieve herd immunity? There's a lot of people who might be hesitant to take the vaccine. And if they do decide not to take the vaccine, or it becomes too complicated to get the vaccine, it's possible that we don't have herd immunity. So one of the consequences of that would be that SARS-CoV-2 could become endemic. It could be just one of many cold viruses that we get occasionally, and for most people, it would be just another cold, but for other people, it would be lethal, just as it is now. And we might have to keep having our masks and social distancing forever, because we just have clusters and pockets of COVID-19 popping up here and there and just in different locations. Another risk, if we don't have herd immunity, is that we can have more novel strains emerging. You can have what's called antigenic drift, where it goes away from the vaccine strain, and then we have to have updated vaccinations. And quite worryingly, it's possible that it could jump back to wildlife. We're thinking this virus came from bats. We have bats in North America. Those are probably susceptible to this virus. And once it's in the wildlife, there's really no way to get rid of it. Then we will have it forever, because it can always jump back from bats to people in North America. So these will all be pretty bad. So I'm asking everyone to help us work together to avoid these outcomes and get vaccinated. This vaccine development process, I would characterize it as the Apollo program of our lifetime. It happened so quickly, not because there was something wrong, but because so many people put so much effort and energy and money to make it possible. And you know, the way that we can honor that is that we do take the vaccines and protect each other from infecting each other with COVID. If you're interested in anything COVID and statistics, I'm teaching a biostatistics class in the spring, and I'm also teaching my disease ecology capstone. And I'm also working on a project with students figuring out how to reopen Eastern by the fall under all these uncertain conditions. Okay, that's it. Thank you.
Okay. Well, thank you all very much for your presentations. And now for folks, if you have questions, there's the Q&A button that you should see at the bottom of your screen. And if you want to type in questions there, we'll see them and try to work through as many as we can. And perhaps we can get started with Dr. Karlberg's question. So which cells take in viral mRNA and respond to it, and responses, and also how does aging affect the response to the vaccine? So one of our panelists like to address this question. Perhaps Javier could.
I could give it a try. So the mRNA vaccines, as far as I understand, they're in a vesicle made of lipids. The cells take them by endocytosis. Now, I don't think there's a good way to, at least for now, target which cells are taken. But I guess the area of injection will be the natural area where the cells will be taking these liposomes with the mRNA vaccine. And then how do they know that? Again, unless we know how to target specific cells, I don't think they know that. So they will be taken by those cells that are able to endocytize the liposomes. At least that's how I understand it. Now, I can't see the question about age in Justin. You mind reading it? I can't find it in my.
How does aging affect their response to the vaccine?
Yeah,
so that's over to the answer
question. So as we all know, the immune system changes while we age. We don't have the capacity to create or generate immune cells as we used to when we were younger. So I guess it's all at this point speculation because we don't really have, or we had not the time, we don't have the time, we have not had time to evaluate the impact of the vaccine in this patient or aging population. But my guess would be that not knowing the road or the way it's going, it's definitely going to be different. However, vaccines seem to be effective against severe cases of COVID, and aging populations are the ones that are suffering the most severe cases of COVID. So my hope is that even though those responses are not as effective as they would be in a younger population with a stronger or fresher immune system, they would still work. But I think that the responses will be different for sure. Yeah.
So we have a lot of questions have come in now. Are there, I can continue picking them, or if any of our panelists have particular questions they're excited about answering, feel free to jump in.
I can answer Chris's question really quickly. So for the AstraZeneca vaccine, has the gene for the spike protein been cloned into an adenovirus? And so yes, there is a specific adenovirus from monkeys that works quite well because we don't actually produce an immune response against it. And so we basically took out all the pathogenicity genes anyway and just have enough information so the virus can get into the cells and can begin transcribing the mRNA for the spike protein, and then the cell translates it, but the virus doesn't do anything else. And with Karen's question, with Dr. Carver's question, so the dendritic sites, the dendritic cells are the hopeful target. And it does seem that the phospholipid, the combination of phospholipids that they use can actually, and it's not exactly clear how, but can actually sort of bias towards some of the cells. And then they're brought in by usually clathrin-dependent endocytosis. And that's probably why both Moderna and BioNTech patented so many lipids that it's a huge list of lipids that gives them the ability to just make cocktails and try different liposome compositions.
So we have a question on the difference in vaccine development between SARS-MERS vaccine and COVID-19, and what has changed that allowed us to develop effective vaccines to COVID-19 so much more rapidly. And there are panelists who'd like to.
Money.
I would also add that MERS is very rare. Like, you know, it's probably less than, I don't remember exactly, but it's on the order of maybe a couple of hundred people had MERS, like, ever. And so it's very difficult to do a vaccine trial on a rare disease, because you have to enroll a sufficient number of people that there could be a statistically significant difference between the number of people who do get sick in the control group and the treatment group. And with a rare disease, you have to enroll tens of thousands of people, or maybe hundreds of thousands, and then wait. Yeah, it's just not feasible for a rare disease.
I guess what I was, tongue in cheek, but we could develop vaccines against almost anything if the resources were there, and we could do it this quickly if the resources were there. It's just for this, it's a big problem, and there were lots of resources put in, which is why we can develop a vaccine so quickly. We've known how to make mRNA vaccines for over 10 years. It's just now, there's billions of dollars going into these companies.
I would like to respond to the question on the masks in general. So I think it is a societal shift for sure. In Southeast Asia, it has been a much more widespread use of masking previous to this, especially since the original SARS outbreak. And it's almost a courtesy if somebody is infectious with anything, even the common cold, they would wear a mask to protect others. We haven't had this in the US. We did have mask wearing during the Spanish flu, but then it sort of disappeared. And one interesting thing is the mask wearing is having a major effect on other respiratory diseases. Basically this winter, we have very little flu, if you noticed, very little common colds. I haven't been sick once since we've been teaching from home. And so honestly, mask wearing is just generally very powerful in preventing respiratory infections. So even once I have the vaccine and we don't have to worry about COVID anymore, I might just keep wearing it just so that I don't get the flu.
Well, Dr. O'Quinn has asked a question that I have been wondering about a great deal as well, which is, if we are actually given the choice, which of the available vaccines would you recommend? And why?
Well, go ahead, Luis.
So, I mean, you can, this is an opinion, so I think everybody could have an opinion. I personally would take whatever vaccine is available. With that being said, if I had to choose between the Mrna and the Pfizer, I would probably take the Mrna one because you have similar results and there's less stuff in the vial. With that being said, all of the vaccines are safe. And so I would gladly take whichever vaccine is in the needle next to my arm. As soon as they want to give me one, I will take it.
Same here. I'll take the first one.
Agreed. The one dose is going to be more convenient, but I'll take the two if that's what I can get.
One thing I read on CNN just this morning by Michael Osterholm, one of the leading experts, is he said that he would, if there's only the Johnson & Johnson, he would take that. And then at some point later on, he could still take the Pfizer.
So we have a question on basically, would anyone like to respond to the comments from the general public about how do we know that there aren't long-term negative effects of the vaccine? How long does it normally take to verify that there aren't any long-term negative effects? Have there been vaccines for which we've discovered long-term negative effects sometime after they've been approved?
So there have been some cases of vaccines that had to be redesigned. The polio vaccine is the clearest example. I mean, there were years after the vaccine was approved until some cases of the virus actually emerged in a very small population, but still a fraction of the population that was very significant, even if there was one case was significant. So there have been cases of new vaccines being developed after approved vaccines have been in the market. Now, how long do we need to wait? Well, yeah, I don't really have a good answer for that, unfortunately.
I mean, the polio, Javier obviously is totally correct. The polio vaccine was an attenuated virus, as was, the smallpox vaccine. And I was actually still as a kid vaccinated for smallpox and ended up with a big welt on my arm. But we aren't really doing attenuated vaccines very much at all. And so all you're really getting is a small piece of protein, and all of the components have been deemed safe. And so if I was a betting man, we really shouldn't see any long-term effects from this vaccine. And we know for a fact that we're going to be seeing long-term effects of a COVID infection. We're already seeing some cardiac issues and things like that. And unfortunately, so we definitely need to get everyone vaccinated.
Yeah, no, definitely. There's no spontaneous generation here. We have a protein. We have a tiny fraction of the virus itself, nothing else that is being injected. But not even that in some cases with the mRNA vaccine, it's not even the protein, it's the mRNA.
Right, and it's not even, the mRNA doesn't even code for the full spike. It's just a portion of the spike. I actually was not able to determine how many epitopes they actually did, but I suspect that they found some highly immunogenic epitopes and focused on that.
I agree with Luis that the calculation between the risk of the vaccine and the risk of the disease is just, like magnitudes different. Like the number of severe cases, anaphylactic shock was maybe like 6 out of 30,000 for one of the vaccines, and anaphylactic shock we can treat. That's why people sit for 15 minutes after they get their vaccine. So if somebody does develop anaphylactic reaction, they have EpiPens on site that they can easily treat it with. Whereas the risk of severe symptoms and deaths from COVID is magnitudes higher than that.
I would answer Lynn Hardy's question. That's all right.
Yes.
So Lynn Hardy asks, as a member of the interested general public, I have read that viruses that target RNA are more prone to mutation than viruses that target DNA. So I guess first I would sort of restate that. So we have viruses whose genetics are RNA, or viruses whose genetics are DNA. And so the viruses that have DNA as their genetics, their genome is usually replicated by our own enzymes. And our enzymes are very good at not making mistakes. So the DNA polymerase that replicates the genome of a DNA virus is the same DNA polymerase in your cell that replicates your genome. And so it's over evolutionary time, it's become quite efficient through the proofreading process. Whereas for RNA viruses, the actual polymerase is very error prone. And so it makes a lot of mistakes. And that's why we're seeing all these variants, is because we have lots of replication. Lots of replication leads to replication errors, and those can accumulate. And then through the selection process, some of them that are more successful, which is not good for us, right? That they can spread better, but if they can spread better, they're more successful, so they become more common in the populations. And with that, basically, just as in case somebody might ask, so these new variants, what it looks like is that they can actually attach to the ACE receptor more tightly. And so there's a higher probability of getting into the cell if you can grab on with more strength, basically. And so looks like that's what's going on there.
I had a question for Krisztian, actually. So in terms of our herd immunity calculations, of course, there's currently a lot of people who aren't approved to take the vaccine, most conspicuously people under 16. So what does that mean in terms of those who could take the vaccine? What percentage of them do you know offhand? Presumably that means we have to get a larger percentage of the people who are actually approved so far to take the vaccine.
As far as I understand, there are continuing clinical trials and that do enroll children below 16 years of age, because eventually we will need to get our kids also vaccinated. It's just we do know that this particular virus causes less serious symptoms in children, and therefore they are at lower risk of severe symptoms, and that's why we don't prioritize them at the moment. But in order to achieve herd immunity, I think it will be critical to also vaccinate children below 16. I don't know if that, how young that will be possible, because there are some vaccines we don't administer below 6 months of age, like the measles vaccine, or even 1 year of age. But until then, it just means that, the rest of us who can get the vaccine, it's even puts more responsibility on us to enough of us to get vaccinated so that we can achieve herd immunity despite all our kids not having the vaccine yet.
And well, maybe a good final question. A couple of the questions that people have entered have been along the lines of, do you have suggestions for dealing with people who are vaccine hesitant for whatever reason, or who simply refuse to follow guidelines in terms of keeping everyone safe during our current pandemic? Aside from the science, questions on, I guess this would be communication.
I mean, I would say that you need to meet people where they are. You can't, other than legal requirements, you can't really force them to do anything. And if people are hesitant about taking the vaccine, my first question would be to understand why they are hesitant. What is the issue that concerns them, and then try and address that issue specifically. It usually doesn't work to just bombard people with information. People tend to become resistant to, perceived pressure. So it's better just to have a discussion and just recognize that you won't be able to convince everyone. There will be people who have such set belief systems that any fact, no fact is going to override. I'm interested in what the other panelists think of that question.
I'd say we're all in this together. If you're not doing these things for yourself, do it for the people that you care about. Do it for the businesses that you care about. Wearing a mask isn't fun. Running a fever after getting vaccinated isn't fun. But if we don't pull together and do it, this is our world now, and this is how we live forever. So the sooner we all get on board, the sooner this ends.
I totally agree. It's a social question instead of a scientific question. And I guess I would say, you know, hopefully we did a good job today and you can show them this presentation and the science will prevail.
Nothing to add, really.
Okay, well,
Justin,
with that, yes.
There was a question about sharing the presentations. I mean, I'm totally okay with that. So I don't know if we have an idea of how to do that.
Yes, so there's going to be both a recording of the seminar today, and then if speakers would like to share their slides, for those attendees who are within the biology department, we can distribute it through the biology canvas. Let's see, for those who are not in biology, feel free to email me.
You should also be able to put it on YouTube. They have a department YouTube page.
Oh, that's a great idea, Krisztian. Thank you for that. So once we get a hold of the recording, I'll figure out how to get that on YouTube as well. But if you can't find it and would like access to it and you're not in the department, feel free to email me. I'm jbasto@ewu.edu. And just a heads up, it takes a few days to get the recording from IT. So it will be a few days before that's available, but we will make sure that's available. And thank you all so much for coming. And again, a big thank you to our presenters. And I hope to see you all again in a couple weeks.
Thank you all.
Thank you.
Thank you.
Thank you.
Hey, Justin.
See you over in the faculty meeting.
Yeah.